Okay, let's continue our series on genetics, grade 12 genetics. We ended the previous video answering this problem, and we'll be answering the next one. So this example is an example of a dihybrid cross, so dominant recessive, or in other words, sex-linked. So you are given an instruction to cross a female. Let's define first the two traits, basically the two main traits that we'll be having. Trait types. So we have in terms of height and in terms of whether or not the individual has hemophilia. Hemophilia, I believe, is an X-linked recessive inherit trait. That's the reason why hemophilia is classified with the letter small h instead of having the big H, because again, the trait is known to be recessive. And for in terms of the height, they say that being tall is the dominant trait and being short is the recessive trait. So what we're supposed to cross is a female who has, we can't say that she is hemophilia positive. She is basically a carrier. If she has hemophilia, there has to be another H, like in the exponent for position, if this X is a constant. We're talking about what's going on in terms of math or algebra. So we have two capital T's over here. So that means this is a tall female carrier, and she is homozygous. And the other individual, the male, is said to be short because of the TT. How do we know that whether or not someone is homozygous or heterozygous? Well, with our female example, we have two capital T's. Instead of having rather capital T small t, this is supposed to indicate that the individual is heterozygous, but that's not the case. It's homozygous if what you have is this or this, in the context of what we have right now, in this kind of dihybrid cross example. So let's talk about the never mind. Let's try to spread this entire thing in a Punnett square. I could actually just talk about how it's done, but you know what? Let's try actually doing it. But anyways, the square is over there already. It's already written, but let's just start from scratch again. This is something that I wasn't able to do in the previous video. Was kind of on the rush when I was making that video. But let's do it here. So in algebra, we know something called the FOIL method. This is something that we could also use in genetics, in making Punnett squares. Granted that we have a dihybrid cross problem. So basically, what we do is something like this. We have XHT. Next is XT, I believe. Is it? Okay. So first is going to be this, and the next is that, which is essentially the same as the first one. The third one is going to be XT, and the fourth one will be XT as well. Yeah. So it's the same thing. Um, why is it here? And why is it that in my square right now, it's in another side? Well, it doesn't really matter where you put it, as long as you designate, for instance, in this Punnett square, we know that this is for the female, so we put a sign like that. That goes for the female, and the rest, the ones over here, define what is for the male. So here we'll just say that this side is for the female, and the other side is for the male. So xt, and then another xt, yt, and another yt. Okay, so far so good. As much as possible, especially when you're dealing with the dihybrid cross, a reasonable amount of space, because eventually, I'm not sure if it's mandatory for all occasions. It's usually protocol for teachers to tell you to write like these stuff. So basically, you interpret whatever is said. Um, so for example, here you have h and then x and then capital T small t. So what does this mean? First things first, this is a female, so you write that symbol for a female. Um, it is heterozygous, but what matters is that the dominant trait is followed, so therefore we describe it as tall. And since we have an H over here, I'm not sure why I already classified it as hemophiliac. Why I classified the female as hemophiliac when in fact she could possibly be a carrier, not someone who is already completely infected with hemophilia. But anyways, yeah, I believe this is already hemophilia, so let's just get on with it. Okay, back to where we are. Okay, where are we? Okay, let's start filling out the boxes. So xh, x capital T small t, xh, xt small t, xhyt small t, xhyt small t, xhxt small t, xh x capital T small t. Again, we're looking from these two. xhyt small t, xhyt small t, xx capital T small t, xx capital T small t, xy capital T small t, xy capital T small t, xx capital T small t, xx capital T small t, xy capital T small t, xy capital T small t. Okay, we're finished. Now let's describe them. Okay, here we have a female who is hemophiliac, and she is tall. Yeah. Next, we have another female. She's also tall, and she has hemophilia. I suppose since this is also known to be like basically the female is a carrier of whatever trait that is. In this case, it's hemophilia. I suppose it applies in the context of working on pedigree diagrams, but that would be for future videos. I'm not sure why here it's not considered carrier. Or maybe it is. I'm just not aware about it. Okay. Sorry about that. This is supposed to be the symbol for females. This one is for male. Okay, we have tall, and then yeah, this guy has hemophilia. Does he? Let me confirm. Yep. Must be. You only know that the individual is normal regardless of sex if he or she has no H exponent. What is this? Oh, okay. Next, this is a female. She's tall, and because there's an H, that means hemophilia. This one is a guy. He's tall, and he has hemophilia. This is also a guy, tall, and with hemophilia. Next, this is a female. She's tall, and she's normal. There's no H exponent this time. That also goes for the next. As you can see, it's literally the same writing. For the other two, they're all tall and normal. The only difference is the sex. There you go. Next. Oh yeah, essentially they're all the same. These two below. And yeah, these other two at the right follow suit from what's given from above. Okay, we're now done. Now let's go find out the genotypic ratio. But first things first, let's list down all of the symbols, combinations of symbols that are present in everything, in each square, basically. So we have x raised to h, and then x capital T small t. So let's try to highlight them. So we have this, what else? This and this. So I think we have 4 of them. 4 out of what? In a dihybrid cross type of Punnett square, how many boxes do we have? So 4, 8, 12, 16. We have 16 boxes. So 4 over 16 is 1 over 4, in other words, 0.25 or 25%. We'll be writing all of what we will be writing right now in an actual ratio. Just right now, I prefer writing it into percentage. So next, by the way, this is the 25%, just to avoid confusion. We have another x raised to h, and then y, and then capital T small t. That these 4 correspond to this. So again, 4 over 16, 1 over 4, that's 25%. Next, xx capital T small t, and then we have xy capital T small t. I believe this, and this belong to another 4 over 16, and that's another 25%. And yeah, I believe the other remaining 4 over 16 corresponds to another 25%. And okay, that's it. genotypic ratio. Overall, uh, let's say 4, 4, 4, 4. But this kind of ratio could be simplified as 1, 1, 1, 1. So essentially, this is the genotypic ratio. What is the phenotypic ratio? So first things first, let's list down all of the names of the symbols that we just wrote. Um, if the genotype refers to the symbols, the phenotype refers to the manifestations or the meanings behind those symbols. In this case, hang on, let me just fix this. Really long. Okay. Phenotypic ratio, we have, I believe we have 4, following what we organized for the genotypes. So we have female, tall, hemo. Let's just abbreviate hemophilia as hemo. We have female, sheesh, why do we keep mistaking the symbols? This is supposed to be for females. Big sorry. Okay. Female, tall, normal. We have male, tall. I think it's safe to say that everyone is tall. There's a 0% chance of for an offspring to be short based on the circumstances presented by the Punnett square. Tall what? Sorry about it. Tall hemo. And then male, tall normal. Again, I believe the percentages are the same. You might ask why I didn't highlight. If the genotypic and the phenotypic ratios, if the findings are essentially just going to be the same, like I could have just highlighted this entire thing instead of just this. This is just to be safe because it doesn't, this kind of scenario doesn't apply all the time. There are circumstances wherein I believe we've already encountered something similar to this, similar to what I'm describing. There is a scenario wherein, yes, you do have the same phenotype, but the symbols are written in a different way. So in that case, that's when the genotypic and the phenotypic ratios are different. So yeah, just to be careful, you want to highlight um, the genotypic ratios. You want to distinguish them from the phenotypic ratios, even though there's a tendency for them to be the same. So yeah, let's highlight this 25% female tall hemo gray. So yeah, we have the same thing. Next color, let's try this. Female tall hemophilia. How about the I'm running out of colors. No, no. Female tall normal. It's that. How about the female tall? This is a bad highlight. Female tall normal. So that's that. And the phenotypic ratio, it's essentially the same as the genotypic ratio. So that's it. Do we have the same? Yes, we do. So genotypic ratio and the phenotypic ratio are the same. That's it for this problem. Let's move on to the next pages of my reviewer. Patterns of inheritance, molecular basis of inheritance, uh, types of asexual reproduction in plants. Okay, so let's cover this one first. So, what are the patterns of inheritance? We have pleiotropy, polygenes, epistasis, codominance, incomplete dominance, and I believe multiple allelism. In the previous video, I did mention something about codominance, incomplete dominance, and possibly multiple allelism as well, based on certain problems regarding Punnett square. So let's start first with pleiotropy. So what's pleiotropy? This is basically the production of a single gene of two or more unrelated effects or phenotypes. So for instance, um, one hallmark example for pleiotropy is something about cystic fibrosis. This is a disease as well as sickle cell anemia. So with just one gene, this could result in many physical symptoms or manifestations. Pleiotropy is recognized to be the opposite of polygenes, which is our second pattern of inheritance. So what is polygenes? What is the meaning of polygenes? So this is basically when multiple genes affect the same phenotype. So an example for this is eye color. Eye color is determined by many genes. For example, OCA2 and HERC2. You don't have to know these genes. But yeah, essentially, for an eye color, a single eye color, many genes dictate what it is. And yeah. So, how about epistasis? Epistasis, this is the interaction of non-allelic genes. What describes a gene that's not allelic? So, there are different loci. Uh, the, the chromosomes have different loci and they have the same phenotyping. That's what it means to describe a gene as non-allelic. And yeah, epistasis is the suppression of the effect of one gene by another. So one example for epistasis is the gene for baldness. So there is, let's say that you have red and brown hair genes, but because of the baldness gene, these red and brown hair genes are suppressed just because you have baldness. Baldness runs in the family. So something like that. Let's talk about codominance. So codominance results when two alleles controlling a trait both express themselves in an organism. So yes, in the example that I talked about in the previous video, it was about chickens, I remember. So black and white chickens, and if you try to cross a black and white chicken, there's a chance for the offspring to have a checkered color. So the offspring basically expresses traits coming from both the mom and the dad. So black and white, checkered. And yeah, it also applies for the case of roan fur in cattles or cows, basically. Um, by the way, allele is basically the different variations of a gene. Okay, next pattern of inheritance, incomplete dominance. It's somehow similar to codominance. It's easy to memorize in the sense that um, instead of expressing both traits in an organism, incomplete dominance is recognized for resulting in a third phenotype. So let's say a flower is red and it's crossed with a flower that is white. So there's a chance for the offspring to be pink in color. And this pink is the third phenotype. So what is incomplete dominance basically? It's one allele for a specific trait. It's basically not completely expressed over its paired allele. It's a form of intermediate inheritance. Okay? Next, we have multiple allelism. So the gene exists in more than two alleles. Okay, kind of makes sense, I hope. So it's recognized that IA is dominant. We're talking about blood types, by the way. IB is dominant, and then a small I means it's recessive. So um, there's a table. So for the genotypes, if you have IA, that corresponds to blood type A. IA and then I, that's also the same. I think that's what this means. So the gene of blood type A exists in more than two alleles. Alleles refer to this, I suppose. I think I'm wrong. I think I'm right. I'm not sure. You can confirm coming from the internet. Just search. Uh, for blood type B, we have IBIB and IBI. For blood type AB, we have IAIB. And for blood type O, we have II. So yeah, I won't describe this or explain it anymore. Next, genotype. What is genotype? This is the genetic makeup of an organism. The genetic makeup refers to the combination of genes in an organism, and this organism contributes to the phenotype. Uh, yeah, phenotype. I have no description for that, but again, what just like what my biology teacher used to always say, phenotype is basically the physical manifestation instead of the symbol itself. I've mentioned that like for the third time already. Okay. Polyploidy. So what's polyploidy? This is when an organism has extra sets of chromosomes. So back to when I discussed about genetic anomalies, like for example, Klinefelter syndrome, Patau syndrome, Down syndrome. It's somehow related to this in the sense that all of the genetic anomalies I've mentioned in the previous videos, they basically fall under the category of aneuploidy. Um, I know we also studied or determined which between these two types are more dangerous. I think it's aneuploidy. I'm not sure. Might as well confirm using the internet. But yes, back when I was preparing for an exam corresponding to this lesson, I mastered all of those topics. It's been some time. It's April 28th. I prepared for the exam for this back in mid March. Anyways, polyploidy. Um, if there are three sets of chromosomes, then it's a triploid. If there are four sets, then it's a tetraploid. But essentially, when we say poly, it can be more than one set or two. Just like how we label polynomials, we could say that we have um, tetranomials. I think that's what they call it. If the highest exponent is 4. But essentially, after trinomial, we call it polynomial. And that's basically a universal term referring to ones with exponents of 4, 5, 6, and above. Anyways, aneuploidy. Aneuploidy is basically chromosomal aberration. One or more chromosomes are present in extra copies or they are deficient in number. So, what's an example of an extra copy? I believe Down syndrome is trisomy 21. In other words, the addition of a chromosome 21. In the case of having a deficiency in a particular chromosome, I hope I still remember it. Do I still have it here? I hope I do. Where is my other reviewer? Is it here? Wait, I don't think so. We left uh, at page 31. Let's get to that later. Yes. An example for a deletion kind of aneuploidy is Credo-Chat syndrome or cat's cry syndrome. Deletion of chromosome 5, basically. Okay, back to where we are. 31. Okay. Okay, let's move to chromosome. Chromosome is a rod-like structure. Why am I? Why don't I see my blinker? I can't see it. Frick. There, found it. Okay, chromosome is a rod-like structure visible in the nucleus during cell division. It's essentially made of DNA molecules. So this is a chromosome. The telomere is over here, and the DNA is basically what composes a chromosome. And yeah, this is a typical cell, and the nucleus will always be inside the chromosome. The chromosomes are inside the nucleus. So as you can see there, those X's, it's basically this one in a smaller form. And what is a gene? The gene is the unit of inheritance born on a particular locus of a chromosome. So adding to that, what is the ranking in terms of size? So cell is the largest. The next is chromosome, then DNA, then what's smaller than DNA, we have protein. By the way, something about the history of the DNA helix structure model. Um, I'm not sure if it was Rosalind Franklin or Watson and Crick who came up with the model, but essentially, it was thought that Watson and Crick were the ones who first discovered about something about DNA, about the picture area something, but it was supposed to be Rosalind Franklin who'd be getting the recognition she deserved. Anyways, molecular basis of inheritance. We have purines and pyrimidines. Um, A and G refer to purines. C and T refer to pyrimidines. Purine plus purine is too wide. Pyrimidine plus pyrimidine is too narrow. Purine plus pyrimidine is with consistency. So this is best. That's the reason why if you match purines and pyrimidines, for example, A from here, from the purines, and T from the pyrimidines, AT, they match. They're complementary to each other, essentially, as well as for G and C. Now, don't even ask why it's not allowed to do like AC, because I'm also not sure why. Okay, next. This is a lot. Okay, SRY gene. SRY gene is a Y-linked gene. It triggers male sexual development pathway in animals. So this doesn't cover humans. The SRY gene is found in males only, because again, males have frick an XY. Females have XX. So because it's a Y-linked gene, it doesn't relate in any way to females, only males. And yeah, if affected, organism is still male, but organ development abnormality is increased in terms of risk. How about bar body? So normal somatic in cell nuclei of female mammals. Okay, so this is for females. Dense body formed from a deactivated X chromosome. This moves near the nuclear envelope. So Bar body indicates organism has more than one X chromosome, and the function is to exist in an inactive form to mitigate gene dosage effects. I don't get this, but that's what I wrote anyways. Homologous chromosomes. So what does it mean by homologous? We already know what a chromosome is, but what does homologous mean? So regarding chromosomes, it's basically chromosomes with the same structure in terms of size and shape, as well as the sequence of genes. So yeah, I like my drawings. Okay. What's hermaphroditism? Uh, in other words, intersex. So organism with both male and female reproductive systems. Okay. Next, we have chromosome translocation. Translocation, reattachment of a gene segment to a non-homologous chromosome. So non-homologous means not of the same size and shape. So what's an example of translocation? I believe CML, or chronic myeloid or myelogenous leukemia, is an example. Uh, translocation of 4 and 21, I believe. Do I remember it right? Let's go back to the genetic anomalies. This is so useful. Read the chat. Chronic myeloid. Where does it say? Sorry. Chromosome reciprocal translocation between chromosomes 9 and 22. Okay. I got wrong with the numbers, but anyways. Okay, so that's an example of translocation. Chromosome inversion. I don't think there's an example for this in my reviewer. But basically, what we mean by inversion, uh, inverting of a gene, rearrangement. So a segment of a chromosome is inverted 180 degrees, even if the segment stayed in the same position. The result, protein produced, is basically different from if there was no inversion. Okay, let's move on to the types of asexual reproduction in animals. You know what? I'll just make a separate video for this. But let's finish the other kinds of changes that can cause a genetic anomaly. So we have chromosome deletion. Deletion example again is to create the Chat syndrome, deletion of chromosome 5. How about duplication? Trisomy. So I believe Down syndrome is an example. So trisomy 21, literally from the word trisomy. So there's an addition of a chromosome 21. So that's what Down syndrome essentially is. Okay? Next video will be about the types of asexual reproduction. And yeah, another video will be on starting slide 35 of this reviewer. Thanks for watching.